Okay, I hope you didn't get too excited when you saw that part one was only about 8.5 minutes long. I had some technical difficulties on my previous recording and I had to shut it down early. So here we go with part two of cellular energetics. This is AP Unit 3, and roughly I'm covering pages 126 to 132 and also page 141 in the Campbell Biology in Focus textbook. And when we left off last time, we were talking about cellular work. And already we have talked a bit about a molecule called ATP. So what is ATP? Well, ATP is an acronym that stands for adenosine triphosphate. And here's a nice detailed picture of what it looks like and what the structure is. So ATP is made up of three different types of components. You have a 5-carbon sugar called ribose. This is the very same 5-carbon ribose that's found in RNA molecules. We've talked about that before. You have a nitrogenous base, a purine base called adenine, the very same adenine that's found in both DNA and RNA. And the third type of component is phosphate. These are phosphate groups. Remember, a phosphate group is a phosphorus atom bonded covalently to 4 separate oxygen atoms. We've seen phosphate groups before. Do you remember where? That's right. It's part of the structure of a phospholipid. Each phospholipid has one phosphate group. ATP has 1, 2, 3 phosphate groups, hence the name triphosphate. Notice that the phosphate groups each have a negative electrical charge. This one actually has 2. That is significant because when you have molecules that have a like charge, like when this negative charge gets close to this negative charge, they don't like each other. They don't like to be near one another. So it takes quite a bit of energy to force those 3 phosphates to stay that close together. Because they have the same charge, they're all negative, they don't like to be near each other. And so the covalent bonds that I'm pointing to right here, in between the phosphates, they actually have quite a bit of energy stored within them. So just a quick recap of what we just talked about. Structure of ATP: 1 ribose sugar, 1 adenine nitrogenous base, and 3 negatively charged phosphate groups that are held together with high energy covalent bonds. And remember, they don't like being near one another. So those have to be strong bonds to hold those negatively charged phosphate groups together. And again, we can think of especially this bond between the second and the third phosphate as being a really, really high energy bond. There's a lot of energy stored there. That's the idea. ATP, its one job is to store a little bit of energy within a cell, energy that can be tapped into whenever the cell needs some energy to do any kind of cellular work. So how do we access energy from ATP? How does the molecule of ATP release its energy? Well, it's a process called hydrolysis. Remember, hydrolysis means you're going to add water to break a chemical bond. So in this case, the way this works is that water molecule is actually going to be added to break this covalent bond right here. The bond is broken. That breaks the third phosphate off of the ATP. So see, now it's no longer attached. And now there's only 2 phosphates on that molecule. By breaking the bond, energy is released. 1 phosphate is no longer attached. And what we have left over is a molecule called adenosine diphosphate, because it only has 2 phosphates left. And it still has some stored energy, but it doesn't have as much. because some of that energy that used to be stored in this bond, some of that energy has now been released, and that released energy can now be used for cellular work. Now, it's important to note this chemical reaction, the hydrolysis of ATP, can happen in reverse. So if a cell has some excess energy that it needs to store, energy can be used to reattach this phosphate back onto ADP. See, it already has two phosphates. If you reattach the third phosphate, you're turning it back into ATP and you're storing energy. Hydrolysis of ATP releases energy, but if you turn around and you add the phosphate back on, you can store that energy all over again. So it's a never-ending cycle. Energy is constantly being released from the burning of fuels like glucose through a process called cellular respiration. So we have energy coming in from cellular respiration, from burning those fuels. What do we do with that energy? We add phosphate back onto ADP. Remember, if you add a phosphate to ADP, what does that make? It makes ATP. So we're storing energy when we add a phosphate. And then later on, when we need to access that energy, we do hydrolysis of ATP. We break off the third phosphate, and energy is released and can be then used to do cellular work. When you break off the third phosphate, you have ADP. And then later on, if you have some more energy coming in and you want to store it, you just add the phosphate back on. So we can release energy, store energy, release energy, store energy. And that oscillating cycle is happening continuously in your cells as long as you're alive. Not just your cells, but the cells of all living organisms. So we add phosphate to store energy, we release a phosphate to release that energy. Okay, so to understand how energy is processed inside cells, you need to understand a little bit about what a physicist would call thermodynamics. And specifically, you need to understand the difference between an exergonic reaction and an endergonic reaction. So let's first discuss what we mean by the term exergonic chemical reaction. This is a chemical reaction in which the reactants, the chemicals that we start with at the beginning of the chemical reaction, have more energy, more stored energy than do the products. So that means somewhere during this chemical reaction, energy is output, energy is lost. Now, in physics, we express that as a delta G value that is negative. We call that a negative delta G. That means negative change in energy. So I know that's a little confusing, but let's look at a specific example. And this is the example of aerobic cellular respiration, where we have a high energy glucose molecule. Glucose has a large amount of stored energy. We use oxygen to break down the chemical bonds in the glucose, and at the end of the chemical reaction, we have products, carbon dioxide and water, which are very low energy. So if you start out with a high energy reactant and you end up with low energy products, that means somewhere along the way energy was lost. That's an exergonic reaction. Here's a quick summary. So if it's exergonic, the products have less energy than the reactants. That's a negative change in energy, or negative delta G. That means energy is released. Now in a chemistry lab, if you're running an experiment in a beaker, you're running a chemical reaction in a beaker, you know if it's exergonic because it'll give off heat. If the beaker starts to heat up, you mix two chemicals together, things start to bubble and fizz, and all of a sudden you can feel heat coming off the beaker. That tells you that's an exergonic chemical reaction. Energy is being released in the form of heat. And even cellular respiration releases some of its energy as heat. That's why when you exercise and your body is burning glucose in its cells, your body heat rises and you start to sweat. It's an exergonic chemical reaction. It's releasing energy. Now, the opposite of that is endergonic. That means somewhere during the chemical reaction, energy is being gained, energy is being input. It means that the products, the chemical that we end up with, has more energy than the chemicals that we started with. So that's a positive delta G. Notice it says delta G greater than 0. Here's an example. This would be low energy carbon dioxide, which can be put together with low energy water. Neither of those molecules has much energy, but look at what you can make. If you're a plant, you can do this. You can make glucose, a high energy sugar. You can also make some oxygen. And of course, this is the process of photosynthesis. This is what plants do every day. Using energy from the sun as an input, the plant can take two low energy chemical compounds and use them to drive the synthesis of a high energy sugar. Thus, it's an endergonic reaction. We're not losing energy, we're gaining energy. The products have more stored energy, or what we call more potential energy, than the reactants did. That's a positive delta G. That means so-called free energy is being stored, and a great example of a biological process that is overall endergonic is photosynthesis. Now, here's another thing to realize. Very rarely in nature does an endergonic or an exergonic reaction exist in isolation. Usually, if there's an endergonic reaction, it's going to get its energy from another chemical reaction that is exergonic. And we call this idea in biology coupled reactions. And here's a couple of examples. All right, notice on this graph, the further down we go, that's the lower amount of stored energy. And then as we go up, there's greater amounts of stored energy. So we're going to start with a high energy glucose. We know that has a lot of stored energy. If we do aerobic cellular respiration and we break down that glucose, follow this arrow, it's going to produce low energy carbon dioxide, water, and a little bit of heat. So that's endergonic or exergonic. Oh, it says right there, doesn't it? Gives it away. Yes, it's exergonic. And you know it is because energy is being released. We end up with less energy, see where we are on the graph, than we started with. We started way up here. But notice this chemical reaction is coupled with another chemical reaction that's going the other way. And what is this chemical reaction? We're taking ADP and we're adding a third phosphate to it. When you add a third phosphate, see, it's going to gain energy and become ATP. That would be endergonic because we're adding energy to convert it from ADP into ATP. And as you know, at some point in the future, we can come in, we can add water to this bond right here. We can break off that third phosphate. That's the hydrolysis of ATP. When you break that third phosphate off, it goes right back to being ADP, but it has less energy. See, energy is lost when you remove that phosphate. So that would be exergonic. And that energy that's lost right there can be used to join amino acids together to build or synthesize a protein. That's called protein synthesis, It's definitely endergonic. It takes a lot of energy to build a protein. So notice what we did here. We have a total of 4 chemical reactions. We have an exergonic reaction coupled with an endergonic reaction. And then over here we have another exergonic reaction coupled with an endergonic. Now, if you go from where we started here, really high energy, and you look at where we ended up here with our protein, which of those has the most energy, the protein or the glucose? Look at where they're sitting. Protein has about this much energy. Glucose has this much energy. So overall, if you look at the process as a whole, it was a downhill series of reactions. Overall, it was exergonic, meaning we lost more energy than we gained. And that's typical in a biological system. That's very typical of living organisms, because there's always going to be some energy that gets lost as heat, and you never really get that energy back. I think that's the second law of thermodynamics. I don't know, it's been a long time since I had a physics class. But that's just a little quick introduction to exergonic and endergonic reactions and the concept of coupled reactions. Okay, and just a quick summary of coupled reactions. It's 2 or more reactions that are closely associated with each other. The energy released from an exergonic reaction is used to drive an endergonic reaction. And there are certainly lots of examples of this in living cells. This is a very common occurrence in living cells. Okay, now it would be, I think, irresponsible to get too far into a conversation about energy in cells without talking about enzymes. We cannot forget about the importance of enzymes, because every single chemical reaction that happens in a living cell is regulated by an enzyme. Enzymes, remember, are proteins that catalyze, they act as a catalyst for chemical reactions. Now, what does that mean? It means enzymes have this uncanny ability to increase the rate of a chemical reaction. Chemical reactions that would take minutes or hours or days or weeks or months or years can happen in milliseconds because of enzymes. So it's important to remember that enzymes are not consumed. In other words, the same enzyme can be used over and over again to regulate the same type of chemical reaction. And the one concept that we may not have discussed before now, but we certainly are going to today, is that the way enzymes really work is that they lower or decrease the energy of activation. And I'll explain that in a moment. But basically they do that by providing a surface area, a physical place where reactants, chemicals, can get together in just the right arrangement to facilitate or speed up the chemical reaction. So if you forget everything that I've said, don't forget this. Enzymes are proteins, and they are proteins that speed up chemical reactions. And you remember how this looks. Every enzyme has a very specific tertiary 3-dimensional shape. And there's a specific site or location somewhere on the surface of that enzyme that's called the active site. It has a very specific shape so that one specific type of chemical can bind to it. And that specific chemical is called the substrate. So in this particular example, we're looking at an enzyme called sucrase, and the substrate is sucrose, which, as you know, is a disaccharide. And so when the substrate binds to the active site, there's a slight change in the shape of the enzymes so that they fit really snugly together. And look at what's happening in this case. A water molecule is being added in order to break this covalent bond in between the two sugars. So, what kind of chemical reaction is this? You know, adding water to break a bond, you should know that that's hydrolysis. This is the hydrolysis of sucrose. And the products which are going to be released are individual monosaccharides, in this case glucose and fructose, which are both monosaccharides. But as soon as those products are released, the active site is empty all over again. It's ready to pick up another sucrose, and the process repeats. And the same enzyme can be used over and over and over again. So there's a concept called induced fit. And that means when the substrate comes together right here in the active site, when those two bind, the whole enzyme changes its shape just a little to make that fit perfectly snug. And that's when the chemical reaction takes place. Now, let's look at this graph. Notice we have reactants here. This is where the chemical reaction starts. It's the same as the other graphs we looked at earlier. So progress of the reaction down here on the x-axis, that's just time. So we're starting the reaction right here, and then over here is where the chemical reaction is going to stop. And then this is the amount of energy that the molecules have. So the reactants, the chemicals that we're starting with, have this much energy. And then here's the chemical reaction right here. It's where we end up. The products only have this much energy. They have less energy. The products have less energy than the reactants. So is this exergonic or is this endergonic? You should know at this point. Is this a positive delta G or is this a negative delta G? We started with this much energy, and then we end up way down here with only this much energy. We end up with low energy products. That means it's a negative delta G. That means energy was lost. So this is exergonic. But I want you to notice something. Even though it's exergonic, even though energy is being lost right here, and the graph is dipping down to show that energy is lost, at the beginning of the chemical reaction, we've got this hump that we have to get over. In other words, energy has to be put in order to sometimes to get the energy out. A good example of this would be if you put firewood in a fireplace. You're ready to have a roaring fire on a cold winter evening. Well, if you throw firewood, which is an energy-rich fuel, into the fireplace, it doesn't just instantly burst into flame. You have to put in some energy. You have to strike a match and put in some heat in order to get the fire going. And then once it starts burning, it will release huge amounts of heat. It will release way more energy than you put in. But nevertheless, you do have to put in energy to get the chemical reaction started. This is called the energy of activation. And all chemical reactions require this, a certain amount of energy that must be put in to start the chemical reaction. Sometimes this energy requirement is so great, sometimes it takes so much energy to get the chemical reaction started that it just won't start. And it makes it very difficult and very slow to get the chemical reaction started. So what is our solution to that problem? How do living cells solve this problem, you ask? With enzymes. So here is the chemical reaction that we just looked at. The black line, just look at the black line on the graph. It has a large energy of activation, so it's going to require a lot of energy to be put in order to get the chemical reaction started. But if we run that same chemical reaction with an enzyme, look at the red graph. We start at the same place, we finish at the same place, but what's different is look at the size of the hump. It's a lot lower. The amount of energy required to start the chemical reaction is much, much, much less. So what enzymes do, the beauty of an enzyme is it lowers, it decreases the energy of activation. That means it reduces the amount of energy required to start the chemical reaction. And that, my friends, is a good place to stop part 2. We'll continue soon with part 3 of cellular energetics. Until then, I hope you have a great day. Oh, before I leave you, I almost forgot. I'm going to start embedding within some of my recorded lectures an extra credit phrase of the day. The first 10 students to email me with the correct extra credit phrase of the day, you can put it right in the subject line of your email. First 10 students to email me with this get extra credit. Today's extra credit phrase of the day: energy for me in the form of ATP. Energy for me in the form of ATP. Thanks, guys. Have a great day. See you soon.